Dear students, now I am going to start Equisetum. So first classification of Equisetum. It belongs to group Pteridophyta, division Arthrophyta. It is commonly known as Spinophyta, and class Calamopsida, order Equisetaceae, and family Equisetaceae. So Equisetum is a pteridophyte. It included in the order Equisetaceae and family Equisetaceae. It is commonly called horsetail. It represented by about 30 species. So it is distributed all over the world. Some common Indian species are Equisetum elongatum, Equisetum arvens, Equisetum diffusum. So they live in a variety of habitats such as open grasslands, river banks, roadsides, railway tracks and hills. Equisetum is a sporophyte. It is a diploid plant. It is a herbaceous climber and climbs over adjacent trees. It grows up to height of 10 to 60 centimeters. So this is a sporophytic plant. It consists of stem, root, leaves. The main stem is an underground rhizome, and it has a nodes and internodes. So rhizome that consists of nodes and internodes. Each node has whorl of scaly leaves. The base of scaly leaves are joined at the node to form a sheath. The node that develops branch buds, and the branch buds grow into underground tubers or aerial shoots. So from the branch buds there is a formation of underground tubers or aerial roots. So the aerial shoots that grows above the ground. In this picture you can observe this is the external morphology of Equisetum, and there is a presence of a rhizome. This rhizome is underground, and it has nodes and internodes, and each node has a whorl of scaly leaves, and the base of scaly leaves are joined at a node to form a sheath, and the node that develops branch buds, and the branch buds that grow into underground tubers or aerial shoots. The tubers can develop into new plant during favorable season. The aerial shoot grows above the ground. So it is positively geotrophic, and it is of two types, that is vegetative shoots and fertile shoots. First one, the vegetative shoots. These vegetative shoots are green and photosynthetic. They are branched. They contain nodes and internodes. A whorl of lateral branches arises from the nodes, and the branches are of two types. See, these whorl of lateral branches are of two types, that are branches of limited growth and branches of unlimited growth. The branches of limited growth are short, unbranched, and contain nodes and internodes, and these nodes are without branches. And second one is the branches of unlimited growth. These are branched and with the nodes and internodes. These nodes are with branches, and the leaves are scaly and isophilous. Isophilous means same type of leaves. So they are present in whorl around the node, both in the rhizome and aerial shoots, and the base of the leaves are joined to form a sheath around the node, and the leaves are non-photosynthetic. So non-photosynthetic means there is an absence of chloroplast, and they give protection to the branch buds. The fertile shoots. So the fertile shoots are achlorophyllous, non-green, and they are unbranched. They contain nodes and internodes with leaves at the nodes. It contains small scaly leaves at the nodes. They terminate into strobilus or cone. It is the fertile part of the stem, and the roots arises from the nodes of rhizome. Next, let us move on to the internal structure of aerial shoot. A cross section of aerial shoot through the internode that shows four main layers, namely epidermis, cortex, pericycle, and stele. These are the four main layers of aerial shoot. The epidermis is composed of a single layer of cells. It has a deposition of silica. The epidermis has stomata. Through this exchange of gases takes place, and each stomata has a stomatal apparatus consisting of two subsidiary cells and two guard cells, and an epistomatal chamber and a substomatal chamber. So in this picture you can observe a vertical section of stomata. There is a presence of two subsidiary cells, two guard cells, and an epistomatal chamber and a substomatal chamber. So this is about epidermis, and next zone is cortex. The cortex lies below the epidermis. It consists of an outer cortex and inner cortex. So cortex can be differentiated into outer cortex and inner cortex. The outer cortex is made up of sclerenchyma and chlorenchyma. So in this picture there is a presence of outer cortex. It has a both sclerenchyma and chlorenchyma cells, and first zone is epidermis. It is a single layered, so it has a single layered cells with the cuticle, and second zone is cortex. It has outer cortex and inner cortex. Outer cortex is made up of sclerenchyma and chlorenchyma cells, and the inner cortex that consists of parenchyma, vallecular canals, which forms an aerating system, and endodermis. So there is a halo region. It is a parenchyma, vallecular canals, and the next zone is endodermis, and after the endodermis there is a single layered pericycle. It lies below the endodermis, and next zone is stellar region. The stele is siphonostele. Here siphonostele means there is a presence of pith. The stele is siphonostele, where the vascular bundles are arranged in a ring around the large pith, and the vascular bundles lie alternate to the vallecular canals. So here the vallecular canal that alternates with a vascular bundles. The individual vascular bundles are separated by parenchyma. So in between the vallecular canal there is a presence of parenchyma cells. So here the vascular bundles are conjoint, collateral, and endarc. So the xylem of a bundle is in the form of two lateral and median group of tracheids, and the protoxylem is disintegrated into a carinal canal. So here the carinal canal is nothing but protoxylem. So it disintegrated into carinal canal. The carinal canal conducts water because xylem is a water conducting tissue. So The protoxylem, it is nothing but carinal canal, so it conducts water. The metaxylem, tracheids have scalariform or reticulate or pitted thickenings. And vascular bundles consist of xylem and phloem. Here the xylem is V-shaped. It consists of tracheids and vessels, and the phloem lies in a radial alignment with the carinal canal. It consists of phloem parenchyma and sieve tubes. So here carinal canal, below the carinal canal there is a presence of phloem, and the pith has a large pith cavity. So it is hollow in structure. So internode or the stem of the Equisetum is hollow structure. So this pith is a air cavity or hollow cavity. Next, internal structure of node of Equisetum. A transverse section of stem passing through the node that shows an outer epidermis and cortex, endodermis, stele and pith. The epidermis and cortex are similar to those of internode region of the stem. However, the vallecular canals are absent in nodal region. And next zone is endodermis. So here endodermis is single layered. It lies below the cortex. It is followed by single layered pericycle. Inner to the pericycle there is a stele. Here the stele is similar to that of internode, and but here the vascular bundles are compactly arranged in the form of cylinder. There is the absence of carinal canals. There is no parenchymatous region between vascular bundles, and leaf and branch traces arise from the vascular cylinders. There is the presence of leaf traces or leaf gaps, and the pith is well developed and it is parenchymatous. It forms a septum between hollow spaces of internodes. So this septum is found between two hollow spaces of internodes. This septum is often called nodal diaphragm, and the node is thus solid. Next, internal structure of rhizome. So here the stomata are absent. In epidermis, stomata are absent, and chlorophyll cells are absent, and the other cells are similar to that of aerial shoot. There is a presence of single layer of epidermis, and cortex is differentiated into outer cortex and inner cortex. Outer cortex is made up of sclerenchyma, and inner cortex is made up of parenchyma cell. There is a presence of carinal canals and single layer endodermal cells, and below the endodermis there is a presence of pericycle, and there is a presence of vascular bundle. It is similar to that of aerial shoot. So next, internal structure of root. A cross section of root shows outer epiblema, a middle cortex and an inner stele. Epiblema is single layered. It produces root hair. In stem and leaf, the epidermis is single layer, same as it is epiblema. It is present outer layer for root, and in epiblema there is a presence of single layered, and it produces a root hair. The next zone is cortex. It is made up of an outer sclerenchyma and inner parenchyma, and the endodermis is two layered, and pericycle is absent. And next zone is stele. The stele is protostele. The xylem is diarch or tetrarch. A large metaxylem is present at the center of the stele, and the protoxylem lies around it. So xylem is diarch or tetrarch. The phloem is located between the protoxylem. Here the pith is absent. Next, let us move on to the reproduction of Equisetum. So, Equisetum is a diploid sporophytic plant. It reproduces new sporophytes by vegetative method. The sporophyte produces gametophytes by asexual method, and the gametophyte reproduces Equisetum plants by sexual methods. And first one is vegetative reproduction. The rhizome of sporophyte produces underground tubers. The tubers detach from the parent plant and remain scattered in the soil. On favorable season, they grow into new sporophytic plant. Next, asexual reproduction. Asexual reproduction takes place by spores. The spores are produced by the sporophytic plant. So, the sporophytic plant that produces spores, from these spore asexual reproduction takes place. The spores grow into gametophyte. It may be male gamete or female gamete. So, Equisetum is a sporophyte. Some aerial shoot terminate into a strobilus or cone. So, the fertile shoots that terminate into a cone or strobilus. The strobilus consists of a central strobilus axis and a large number of sporangiophore. Each sporangiophore has a stalk and a peltate disc. The underside of the disc has 5 to 10 sporangia. So, this is the LS of strobilus of Equisetum. There is a presence of central strobilus axis, and a large number of sporangiophore are attached to the central axis, and each sporangiophore has a stalk and a peltate disc, and these the underside of the disc, this peltate disc consists of 5 to 10 sporangia. So, this is the structure of single sporangiophore that bears a stalk and a peltate disc. It has a 5 to 10 sporangium, and this is the LS of a sporangiophore with 2 sporangia. It belongs to jacket layer. Inside the jacket layer there is a presence of sporangium, spores, and there is a sporangium that attaches to the peltate disc. In the maturity of the strobilus, in mature of strobilus, the strobilus axis that elongates and the sporangia are exposed, and the sporangia ruptures and the spores are released to environment. The spores are similar and it is called homosporous. Homosporous means same type of similar spores. The spores are green, spherical and uninucleate. They are formed by division of meiotic division, or they are formed by meiosis, hence they are haploid. The spore is covered by 4 layers, that they are the perispore or epispore, outer perispore or epispore, middle layer, exospore and endospores. When the spores dries up, the perispore splits into 4 bands called elaters, and they help to entangle the spores. They are hygroscopic and respond to humidity. They act as a parachute and help spores to float in air. They help in the dehiscence of the sporangium, and the spores live for a short period of 1 to 48 hours. They germinate in a suitable substratum into a gametophyte. In this figure, Equisetum A that shows structure of a spore. It has a 4 wall layers: perispore, middle layer, exospore, endospore, and centrally located cytoplasm and nucleus. And in figure B, spore with uncoiled elaters. So elaters that helps for it act as a parachute, and it helps spores to float in air. Next, sexual reproduction. Gametophyte is the gamete producing plant body. The spores that producing gametophyte. The gametophyte, it is a gamete producing plant body. It develops from the spores, and the gametophyte reproduces sexually, and it is haploid and monoecious, because the spores that present inside the sporangium, they undergo reduction division. So these gametes are haploid and monoecious. It contains both male and female sex organs. The spore absorbs water and swells. It divides unequally into two cells. They are larger cell and rhizoidal cell. So in this picture, there is a presence of spore with elater, and the spore, it contains spore wall, and it divides unequally into two cell. And first one, larger cell and lower rhizoidal cell. The rhizoidal cell develops into rhizoid, and the larger cell develops into prothallus. The prothallus divides and produces many cells. The basal cell divide rhizoids, and the upper cell are chlorophyllous. So it develops the upper cell that develops into thallus. The prothallus become branched and developed into gametophyte. From these prothallus, the gametophyte is developed, and the gametophyte develops archegonia and antheridia. So from this prothallus, both antheridia and archegonial gametophyte are formed. The archegonia are found in the basal region, and the antheridia are found in the upper region. This is the picture of a gametophyte. There is a presence of archegonia at the basal region and antheridia found at the upper region. Next, structure of mature gametophyte. So the mature gametophyte is brownish green in color. It is found in shady, moist places. The gametophyte is dorsiventral. So it shows both dorsal view and ventral view. So gametophyte is dorsiventral and lying flat on the surface of the soil. It varies from a pinhead size to 8 mm in diameter. So it varies from pinhead size. It is little bit 8 millimeter in diameter, and there is no subterranean portion in the gametophyte. They are semicircular in outline. Here each gametophyte consists of a cushion-like central portion and erect lobes. There is a central portion and erect lobes. The central portion and erect lobes are rich in chloroplast, and so they are green in color. The margin of the central portion is meristematic. Numerous unicellular rhizoids are arises from lower surface of surface and fix the gametophyte on the substratum. So from the lower region there is a formation of rhizoids. It helpful for the attachment to the substratum, and the central cushion like region consists of parenchymatous tissue without intercellular spaces. So these parenchymatous cells are compactly arranged. The lower portion of the cushion or disc is whitish because of the presence of leukocytes. So it is a leukocytes means there is a no coloring pigment in this portion. So there is a presence of leucoplast. So this cushion is or disc is whitish in color. The upper portion is rich in chloroplast. The lobed region is also called spongy portion, and it consists of thin-walled parenchyma cells, parenchymatous cells with intercellular spaces. From this cushion-like region, there is a lobes, several lobes are formed. So this lobed region consists of parenchymatous cells with intercellular spaces. So cells are loosely arranged. This region is photosynthetic and fertile. So it bears the male sex organ called antheridia at the tips of the erect lobes. So from the lower cushion-like region, there is a formation of several lobes. From this lobe, there is a fertile lobe. From this fertile lobe, the male sex organ called antheridia are developed at the tip of the erect lobes, and the female sex organs are called archegonia. It is archegonia formed at the base of the erect lobes, or sometimes it developed in between the erect lobes. Next, structure of archegonium. So the archegonium is the female sex organ. It is located at the base of prothallus. So it is present below the erect lobes, or sometimes it may be present in between the erect lobes. So it is located base of the prothallus. The mature archegonium is flask shaped. It has neck and venter. The neck is made up of 3 to 4 tiers of neck cells. In this picture you can observe 3 to 4 tiers of neck cells, and each tier has 4 cells. The neck has neck canal made up of 1 to 2 neck canal cells. So it is the neck cells has 4 tiers of cells, and it has a neck canal made up of 1 to 2 neck canal cells. The neck projects above the surface of the prothallus. The base is swollen and it has a venter canal cell and a single egg cell. Next, structure of antheridium. The antheridium is a male sex organ. It is located on the erect branches of the prothallus, and the antheridium is spherical in shape. It consists of antheridial jacket and an opercular cell at the tip region and many antherozoids. And the anterior part of the antherozoid is spirally coiled and has numerous flagella. The posterior part is expanded. So in this picture you can observe many antherozoid. These antherozoids are covered by antheridial cell, and these antheridial cell there is a presence of opercular cell. So from the anterior part of antherozoid is spirally coiled and it has numerous flagella, and posterior part is expanded. So it is the Equisetum is spermatozoid. It has numerous flagella with antherozoid. Next, fertilization in Equisetum. So fertilization occur in gametophyte. So the antherozoids swim towards the neck of archegonium. The neck canal cell and venter canal cell disintegrate and it form a passage for the entry of antherozoids. Only one sperm enters the archegonium and fuses with egg. There's a fertilization takes place and the zygote is formed. Usually many archegonia are fertilized in a gametophyte. So next, structure of embryo or development of embryo. The zygote lies embedded in the venter of archegonium. The zygote develops into embryo and the embryo develops into sporophyte plant. The zygote is diploid cell. It divides transversely at right angle to the long axis of the archegonium and producing epibasal cell and hypobasal cell. The second division is vertical and it forms of 4 cells, and the 4 cell again divide vertically, produce 8 cell arranged in 2 tiers of 4 each. The largest cell of the 4 epibasal cell develop into shoot initial and the remaining 3 cells form leaf initial. So the largest cell of 4 epibasal cell that forms as a shoot initial and remaining 3 cells of leaf initial forms as a leaf initial. The largest of the hypobasal cell form root initial and the remaining 3 cell give rise to foot. The root initial multiplies and develops into roots and the root penetrate the gametophyte downwards and they enters the soil, and the shoot initial multiplies and developed into shoot. The first shoot called primary stem comes out of the archegonium and grows up and it develops node and internodes. The node produces 3 leaves. The primary stem stops growth after producing 10 to 15 internodes. Then a secondary stem develops from the base of the primary stem. It also develops node and internodes. The nodes develops into 1 to 5 leaves. It is scaly leaves. The secondary stem has a stopped growth and a tertiary stem appears from the base. This process is repeated to produce 4 shoots. Finally, shoots grows horizontally and penetrate the ground, forming a first underground rhizome, and the rhizome develops a new aerial shoot of the sporophyte. Next, the life cycle of Equisetum. So, Equisetum that reproduces vegetatively, asexually and sexually. So vegetative reproduction takes place by formation of underground tubers, and sporophytic generation is from sporophytic generation the formation of spores takes place. It is asexual reproduction, and sexual reproduction, it is a gametophytic generation. There is a formation of gametes. From the gamete there is a formation of new sporophytic plant. So the strobilus or fertile portion of the aerial shoot that has a 2n number of chromosomes. These strobilus that produces sporangium, it has 2n number of chromosomes. These sporangium, they undergo meiotic division or reduction division to form a haploid spores. These haploid spores, they germinate into gametophyte. It has n number of chromosomes. So from the meiotic division it undergoes sexual reproduction. So the gametophyte develops both antheridium and archegonium. Antheridial at the tip of the erect branches, and archegonium at the below the region of erect portion. And after the formation of antheridium and archegonium, so antheridium that produces antherozoids and archegonium that produces egg cell. From this n number of antherozoid and n number of egg, there is a fusion of both nucleus takes place during fertilization, and after fertilization there is a formation of zygote. It is 2n number of chromosome. From the zygote, embryo develops. From this embryo, the formation of Equisetum sporophyte plant. It has 2n number of chromosomes. This is the graphical life cycle of Equisetum. Thus completes your Equisetum. Thank you.